Hi, everyone. So we're going to continue with chapter 10. This is continuing with microbial metabolism. This is part 3, catabolism, getting materials and energy. So this is all about the breaking down reactions, and the overall reactions are depicted here in this screen, where we start with glycolysis with glucose as we continue to oxidize the pyruvates. Then all of these NADHs and FADH2s go into the electrolyte, lots and lots and lots of ATPs. And if we're using oxygen as the final electron acceptor, which we do in aerobic respiration, we can end up with water, H2, and then carry those electrons from those carriers into a process. So this is, if we were looking in the mitochondria, this is what those molecules would look like. Again, you see the same kind of carriers, and you see ATP synthase. These same special carriers, these cytochromes, coenzyme Q, etc., all embedded into the cristae of the mitochondria or in the plasma membrane of eukaryotic cells. Pay attention as we watch this video to the oxidation and reduction reactions as we go through this short video.
The center of activity that brings your cells to life is found inside the dynamic mitochondria organelle. The inner mitochondrial membrane is coated with enzymes that catalyze the chemical reactions of respiration, working in sequence to generate the electron transport chain. The first step in the electron transport chain is performed by enzyme complex 1. Complex 1 receives electrons from coenzyme NADH, a substrate produced by the citric acid cycle. The catalytic mechanism of enzyme complex 1 connects two different kinds of reaction. Coenzyme NADH is oxidized at one end of the enzyme, releasing two electrons that hop through the interior to coenzyme Q, which is reduced. Traveling amongst membrane lipids, coenzyme Q carries electrons to the next step in the electron transport chain. The movement of charged electrons through complex 1 makes it bend in shape, transmitting energy for pumping 4 protons across the membrane. The second step of the electron transport chain is performed by enzyme complex 3. The mechanism of complex 3 separates electrons from coenzyme Q, passing one electron to cytochrome C, which is reduced. A complete reaction cycle of enzyme complex 3 transports 4 protons across the membrane. Traveling within the intermembrane space, reduced cytochrome C carries the electron to the final step in the electron transport chain. The destination for electron transport is a molecule of oxygen held inside enzyme complex 4. Reduced cytochrome C delivers electrons that transfer to the reaction center of enzyme complex 4. A molecule of oxygen from the air you breathe is captured, split, and reduced. The separated oxygen atoms accept electrons and pick up protons, creating 2 molecules of water. The substrate providing electrons to enzyme complex 1 is coenzyme NADH, a product of the citric acid cycle. A second supply of electrons for the electron transport chain is from step 6 of the citric acid cycle, performed by enzyme complex 2. Enzyme complex 2 catalyzes oxidation of succinate, releasing 2 electrons, which hop through the enzyme to coenzyme Q, which is reduced. Traveling through the membrane, coenzyme Q carries the electron to enzyme complex 3 of the electron transport chain.
So again, these videos I know are a little bit long and very detailed, but they have really good, useful information. So I understand that you might want to speed through them, but if you have the time, I think it will benefit you in helping to kind of understand what these processes are and how they're occurring. So the elements of the electron transport chain include that MIL or ATP synthase, that protein that is associated with the electron carriers and actually captures that released energy. As the protons travel through it, it turns it like a mill, like a grain mill or something like that, to produce ATP. We also, this is called oxidative phosphorylation. It's the coupling of ATP synthesis to electron transport. Each NADH that enters will generate 3 ATP molecules, and each FADH2 will produce 2 ATP molecules. So we call this process chemiosmosis. This generation of ATP by ATP synthase. So we talked earlier, we saw that during the Krebs cycle, we have what we call substrate level phosphorylation, and that's how we make ATPs at the substrate level during those enzymatic conversions in those processes. And then we have chemiosmosis, which is another mechanism of producing ATP. So taking a closer look, we have the protons on one side of the membrane. Now again, if this is in a eukaryotic cell, this will be occurring in the mitochondria. But since we're talking about bacteria, this is happening in the plasma membrane of the mitochondria. And we have this kind of motor, basically, that we call ATP synthase. And as the protons pass through this channel from higher concentration towards lower concentration, doesn't cost any energy to move that way. As 3 of them pass through, creates enough energy to turn the rotor and to cause this to rotate. And that rotation is what generates the ATP from ADP plus PI. This concentration of hydrogen ions is referred to as a proton motive force, meaning that you have more protons on one side than you have on the other side. And that is a type of potential energy. You've stored the energy of those protons on one side, and as they pass through, they give up that energy into the form of ATP. So again, we're going to watch another video on ATP synthase that's going to show us really how this molecule works, because I think just glancing at a still picture of it doesn't really give us an appreciation for how this moves and how this works.
All life on Earth depends on this tiny energetic molecule, adenosine triphosphate, or ATP. ATP drives biochemical activity inside your living cells and is a key building block of DNA and RNA. Generating ATP for your living cells are mitochondria, electrochemical batteries that convert energy from the food you eat and oxygen from the air you breathe into ATP. So where does your ATP come from? Deep inside your mitochondria, rows of molecular motors generate ATP, the molecule essential to all life on Earth. Enzymes bring together reactants to form a chemical bond, converting mechanical energy into chemical energy. A ring of enzymes work in step, creating 3 molecules of ATP with each cycle. Inside the molecular motor, a rotating axle powers the sequence. The axle is attached to a rotary molecular motor, moved by the force of protons pushing from the other side of the membrane. A difference in proton concentration propels the molecular mechanism. Synthesis of ATP. ATP drives biochemical activity inside your living cells and is a key building block of DNA and RNA.
So hopefully that helps you visualize it a little bit better and understand that it's the hydrogens having that gradient that you form that creates this proton motive force. As those protons pass through the ATP synthase, that's what drives the conversion of ADP to ATP. And that's that conversion of the energy in that way. So again, in prokaryotes, this is happening in the plasma membrane. In eukaryotes, this is happening in the mitochondria. So just keep in mind that the processes are very similar, but the location is different. So just make sure that you understand that. So here's the theoretical yield from aerobic respiration. So starting with glucose and the complete oxidation of glucose into carbon dioxide and water, we can yield 2 ATPs from glycolysis, 2 ATPs from the Krebs cycle, and 34 ATPs from that last step, the electron transport step. The carbon is getting broken down into 6 carbon dioxides. At the end, as you go through the whole process, you can see them coming off, 1 in the transition step and then 2 during the Krebs cycle. We get waters being produced throughout this process as well. So here is the total possible production is 40, but because we spent 2 at the beginning, we say the net gain is theoretical is 38. It's typically going to be a little bit lower, probably in eukaryotic cells, because energy is going to also be expended transporting the NADH during glycolysis across the mitochondrial membrane. But nonetheless, 36 to 38 ATPs is pretty good from 1 glucose molecule. And this is showing where those are coming from. Now, remember, all of these ones coming from the electron transport is based on the NADHs and FADH2s that went into the electron transport chain. And those, of course, were generated in glycolysis and the Krebs cycle. Other products are going to be 6 carbon dioxides, 6 molecules of O2 gets consumed or used up in that process to form water, which we're forming 6 waters that are produced during the electron transport as well as in glycolysis, and 2 get used in the Krebs cycle to result in a total of 6, a net of 6 water molecules. Oops, skip a step. So the terminal step at the very end, just to take just a little bit of a little closer look at that. So this is where is catalyzed by the enzyme cytochrome AA3 or cytochrome oxidase. And what's happened is it's adapted to receive the electrons from cytochrome C, pick up hydrogens from the solution that are being pumped through the ATP synthase, and then they're going to react with water or with oxygen rather, to form the water. And here's the overall reaction here. So you have the two H pluses being pumped through the ATP synthase, the two electrons that are coming from the electron transport chain, plus a half of an O2 to make one water. Bacteria can have variations in the cytochrome system or in the electron transport, which means we can use this to differentiate bacteria in the laboratory. For example, we can use a test called the oxidase test. The oxidase test can be used to test for Neisseria, Pseudomonas, and some bacillus organisms. Klebsiella and Enterobacter grow in the presence of cyanide because they lack cytochrome C. Cytochrome C gets bound by cyanide and it acts as a poison, but since Klebsiella and Enterobacter don't have that enzyme, it doesn't poison them in the same way. So that's another way you can differentiate and use these processes to identify bacteria. The incomplete reduction of oxygen can result in toxic oxygen products, which is why cells need their superoxide dismutase and their catalase. Streptococcus lacks the cytochromes as well as catalase, but they instead have a special peroxidase for their systems. So just keep that in mind. Part of the reason why we care about this stuff is because we can actually use differences in how these processes are done by different organisms to identify them. And then by identifying them, then we can figure out how to treat them if they're causing disease. So anaerobic respiration. This is nitrate and nitrite reduction systems. These are found in E. coli as well as some other organisms as well, but we're going to use this as an example. And here, instead of using oxygen as a final electron acceptor, these can use nitrates instead. So what happens here at the end of this process, you're going to see an enzyme, a nitrate reductase, that catalyzes the removal of the oxygen from nitrate to reduce it to nitrate. So from nitrite to nitrate, or nitrate to nitrite, sorry. And again, a test for this is one of the physiological tests that we can use to identify bacteria. Now, interestingly enough, nitrate reducing bacteria that are found in the gut and in our mouths can actually contribute to pulmonary and cardiovascular diseases because we end up with this production of nitrates, nitrites, and other forms of nitrogen. Those things can damage our blood vessels, and that can result in those kinds of diseases. So we want to make sure that we keep our microbiome, the bacteria that live in and on our bodies, particularly our gastrointestinal tract, starting with our mouths and ending with our anus, in balance. Because otherwise we can have these things contribute to diseases, and we're learning more and more about these. That's one of the hot areas of research, is figuring out what are all these connections. And you're starting to see more people taking probiotics and things like that to try to supplement or replace the bacteria that they have. So what about fermentation? So fermentation, pretty much everybody can do fermentation to some extent, even us. But we're talking about bacteria. So fermentation is done by facultative anaerobes as well as aerotolerant anaerobes. And these organisms can use only glycolysis to produce their ATPs. They can't use oxygen, and they don't have other means to carry out anaerobic respiration. So what do they do? What can they do? So here's what happens. You've gone through glycolysis and you've made pyruvic acid. And now you can go through a couple of different processes. One process here is alcohol fermentation, where you go through a conversion with acetylaldehyde, uh, or you end up producing acetylaldehyde from the pyruvic acid as well as CO2. And that is a way to regenerate NAD plus. So you're able to take the acetylaldehyde, and then ultimately the alcohol are able to take on those electrons and recycle NAD plus so it can go back into glycolysis. On the other side, this is a different kind of fermentation reaction. We can produce a number of different acids, including lactic acid. And as we'll see in a few moments, there's other kinds of acids that can be produced as well. And this is what happens with what we call homolactic bacteria. So one kind of fermentation, a single type of acid being produced. And this is what we do as well, as we make lactic acid. And again, the whole purpose of fermentation, you're not generating any more ATPs in the process, but what you are able to do is recycle the NADH so that you don't run out and it can continue carrying out glycolysis. So NAD ends up being the limiting factor in being able to continue carrying out glycolysis. So let's take a closer look at alcohol fermentation. This is going to occur primarily in yeast as well as in certain bacterial species. And they have the metabolic processes, the enzymes that are necessary to convert pyruvate or pyruvic acid into alcohol. So they do this first through the decarboxylation of pyruvic acid into acetylaldehyde, decarboxylation, removal of a carbon group, or carboxyl group. And then they reduce the acetylaldehyde, right? The NADH gives up its electrons and hydrogens there. To produce ethyl alcohol. And this then enables us to, like I said a moment ago, to regenerate NAD that can then go back to be used in glycolysis. Now, of course, we take advantage of this process all the time. If you drink wine or you like bread or anything that you're allowing fermentation, pickles, lots of things get fermented. There's also acidic fermentation, and these processes can be extremely varied, and we're not really going to talk about them in too much detail because, again, they can be very detailed here or varied. But one would be homolactic fermentation, which is what we do and what a lot of bacteria do. And this is just where, again, they produce lactic acid, and this is a way to reduce the pyruvate into lactic acid and regenerate the NADH back to NAD plus to go back to glycolysis. Other organisms do heterolactic fermentation, where glucose is fermented to produce lactic acid, acetic acid, and carbon dioxide. So you can produce different forms of different types of acid if you're heterolactic, right? Many or more than one here. Things to keep in mind here are again, why do we care about this? We care about this because which pathways and which types of acids are being produced can help us to identify the microorganisms. In addition, we exploit the fact that these organisms can make these acids for us to make these acids for industrial purposes. So to produce acetic acid, to make some of these other products, these other kinds of acids that can be produced by different kinds of bacteria, particularly E. coli. E. coli can have mixed acid fermentation. So again, this is not just for E. coli, but all members of the Enterobacteriaceae family, that big group of bacteria. They're able to make multiple different kinds of acids: acetic acid, lactic acid, succinic acid, formic acid, as well as carbon dioxide. So all of these, and you can see these different pathways here of how they can produce lactic acid, formic acid, alcohol, acetate, etc. And these products we sometimes exploit, again, for industrial purposes, as well as to identify bacteria. We can look for, when we grow bacteria on certain types of selective and differential media, one of the things that they can select and differentiate are those organisms that are lactic lactate producers, or that are lactic acid producers, and things like that. So we use this as a way to identify bacteria and also to understand them. A lot of this fermentation that's occurring in our gastrointestinal tract accounts for the gases that get produced in our in that process. So there's things to think about. So the breakdown of non-carbohydrate compounds. So what if you run out of glucose? If the cell always needs ATP, so it will want to keep glycolysis going at the very least, no matter what is happening, because that's what it needs to keep alive. So proteins and lipids can also be used as sources of energy. So I have here, this is just to remind us what a protein looks like. Lots and lots and lots of amino acids that are strung together. And this is showing us lipid A, which is part of the LPS layer of bacteria. And this is just an example, again, to show us these long hydrocarbon chains that are joined together by these glycerol, these glycerol phosphate groups. So these things can also be broken down into precursors for metabolites and energy. They can't just, this whole big chunk can't enter into glycolysis, but you can see we've got some things that are kind of sugar-like up here. We could break these down and use those to go into glycolysis. And the same if we were to look at amino acids. But in order to do that, we need to break stuff down a little bit in order to get it ready to enter into these metabolic pathways for respiration. So we use different kinds of enzymes. One is lipases. Lipases are enzymes that break down fatty acids that are joined by glycerol, or joined to glycerol. It's a way to break down fats. Glycerol can get converted into dihydroxyacetone phosphate, and that can then enter into step 4 of glycolysis. So you're not entering at the very tippity top, but you're entering a little bit lower down into the process. We can also have what we call beta oxidation. This is the oxidation of fatty acids. So 2 carbons get transferred to coenzyme A to create acetyl-CoA, and of course, boom, you're right into the Krebs cycle at that point. Oxidation of 6 carbon fatty acids will yield 50 ATP, compared to just 38 for 6 carbon sugar. And so why is that? Why is it that a 6 carbon fatty acid can make more ATP than a 6 carbon sugar? And the reason is due to the fact that the acetyl-CoA molecules produced from fatty acids, you end up getting 3 acetyl-CoAs per 6 carbon molecule, instead of just the 2 that we get with glucose breakdown. So you're actually, a 6 carbon fatty acid actually generates 1 additional acetyl-CoA. So it gives you one more turn, so to speak, in the Krebs cycle. And so you get all of those extra ATPs and produce from that process. We can also use proteins instead of, if we need to. And we do this by breaking down proteins using enzymes called proteases. And there's a number of specific proteases, but then there's, this is kind of a generic term. And this is what breaks down proteins into their amino acid components. And once you have them broken down into amino acids, you've got to get rid of that amino group, because amino groups don't belong in the whole cellular respiration pathway. So in order to do that, we remove those amino groups through a process called deamination. And so we have to, looking at this amino acid, we have to remove that amino group. And once that happens, the remaining carbon components, as we see here, can enter into the Krebs cycle at various points along the way, depending on what groups it is. So that brings us to the end of this topic, and I understand this has been a long one. So feel free to kind of fast forward through the videos or to just go back and look at little bits and pieces. And have a good day.